Hi, my name is Lacy
Madeline DeVries.
and we're going to be presenting DNA sequencing. DNA sequencing as a whole. DNA sequencing has transformed our society for the better. It has transformed our society in many ways, but mostly in the biomedical engineering and forensic science field.
DNA sequencing has revolutionized on how we look at the genome and the study of genetics and biomedical engineering. It also revolutionized the forensic aspect by finding the perpetrator by using genomes that match closely to their DNA.
DNA sequencing, the breakdown.
In forensic science, DNA sequencing involves sequencing specific regions of an individual's DNA to create a unique genetic profile. So what they'll do, we'll explain it more on, but they'll take a sample of an unknown sample that they collected as evidence, and they'll use different techniques to elongate and separate the DNA and then they'll upload it into a database to try and find matches to create that genetic profile.
In biomedical engineering, overall, engineers use DNA sequencing to study genetic disorders and develop technologies to detect DNA mutations. So kind of similar to the forensic science field. Medical professionals usually take samples of DNA of patients that they are aware of the identity of the DNA strand, but they compare and look at altered mutations within that DNA. We'll kind of get more into this later on in the PowerPoint, but it's a lot of neurological disorders that they're able to find.
The creation and discovery or foundation or anything like that. The first DNA sequencing method that was fully somewhat complete was developed in 1977, which was called the chain termination method or Sanger sequencing by Frederick Sanger and his team in Cambridge. Sanger sequencing itself is a technique that can provide details about the 4 nucleotide bases in a DNA sequence, including their identities and their order.
Here are some of the people involved.
These people all throughout the years that DNA was first sequenced in 1965 to when they introduced a new technique in 1996. They all created steps that they all built off each other to create new ways to sequence DNA.
We think this is a great representation of the evolution of science and how it is a process. The creation/discovery of the first generation. So with genetic sequencing, there is 2 major dates. There's the first generation and the second generation. Around the time of 1972, various researchers began to adapt their methods in order to sequence DNA.
In the first aspects of developing and adapting their methods in order to sequence DNA, those methods were aided by the recent purification of bacteriophages with DNA genomes. And the next large change that made a large impact was the replacement of 2D fractionation with a single separation by polynucleotide length by electrophoresis through polyacrylamide gels.
The creation and discovery of the second generation. In 1996, a new DNA sequencing technique was introduced called
Parasequencing.
Parasequencing. Parasequencing was later licensed to 454 Life Sciences, where it evolved to the major successful commercial next-generation sequencing technology.
Sequencing itself was based on the measurement of luminescence generated as a result of pyrophosphate synthesis during the sequencing. It's later on being licensed to 454 Life Sciences. It allowed the mass parallelization of sequencing reactions, which greatly increased the amount of DNA that can be sequenced in a run. The technology used in forensic science and biomedical, they use around the same technologies and techniques, which many include short tandem repeat, fragment analysis, Sanger sequencing, snapshot, and capillary electrophoresis, single-strand conformation polymorphism. Some newer technology is used as next-generation sequencing.
On the biomedical endpoint, we have a lot of Sanger sequencing used, but mostly it's closely related in the technology used. It's more or less what they do with the results. So in biomedical, you're going to be testing, you're going to be checking for mutations, you're going to be looking at mutated DNA and the DNA that you have in the patient and trying to come up with something. So while the technology used is similar, there are a lot of differentiations with how that DNA is then used and collected. So what and how has it transformed society as a whole? What is transformed in our society through DNA sequencing is how we look at the human genome and understand how we do it today. Another thing that has transformed is the technology in medicine that has used DNA analysis and treatment as a result of DNA sequencing. So basically, to expand on this a little bit more, we look at the DNA from a biomedical standpoint, and we can predict what mutations are going to happen. And then we can go into preventive medical care, which is a big step on how this has transformed society. So if someone knows that they're going to be more susceptible to this DNA mutation, then they can do preventive care to hopefully elongate their life and potentially, with more research, cure whatever disease they have.
How it has transformed our society? It's mostly transformed it throughout the science, technology, and the medicine divisions throughout our world. It's transformed it by being able to solve cases somewhat easier, by able to match unknown genetic material that we find at crime scenes to figure out if this was from the victim, from someone else, from the perpetrator, and to find a match by tracing ancestry and heritage through people as of today to find out where they are, like as in 23andMe, and how they all do those genetic profiles, and along of like how they use it to figure out with somewhat of like the skeletal remains of people who died long ago to figure out like how they were somewhat, and then diagnosing and treating genetic diseases as they do in the biomedical field.
So now we're going to get into the benefits and consequences of genetic altering, or even genome sequencing between the biomedical field. So the benefits of genome sequencing through the biomedical field enables medical professionals to closely examine a patient's genes for mutations and health markers. It can identify development delays and intellectual deficiencies. So again, like we kind of spoke about in the last slides, you're doing preventive care. You're being able to treat and diagnose more efficiently than symptomatic and other influences determining what the outcome of that disease is going to be. So a consequence of biomedical would be the primary drawback of the whole genome sequencing is the volume of data that it creates. Larger storage capacities and more time is needed for the analysis due to the massive volumes of data generated. This raises the price and lengthens the time needed for analysis for patients, as well as it opens the door to genetic altering and the question of ethics. So to kind of go off of this, the primary drawback would be all of the data that it creates. DNA is not a simple thing by any means. So when you're examining data, you're going to have a large volume of it, which makes it more expensive. If you're talking biomedical, insurance doesn't always see that as first-hand care or first coverage care. So a lot of this it's not so much looked at as medical treatment, more than more medical testing. On that note, I kind of mentioned how it opens the door for genetic altering and the question of ethics. So anything that you're looking at the genome and putting it together or comparing it opens that door into, okay, well, how far can you go until there's a problem with bioethics? So this has to do with what if people decide that they want to genetically alter that DNA? What's ethical about that?
The benefits and consequences of the forensic science aspect. There's many benefits that come with the forensic science field with this, which includes increasing the ability to differentiate individuals with using the unknown DNA that's sequenced, generate data that can span the human genome, which can address a wider range of questions, and it can reduce the cost and time by using next-generation sequencing, which can sequence many fragments at once. The consequences of using forensic science with using DNA in forensic science is that the most major ones are the ethical issues with privacy and the consent discrimination due to how many people are in the database and like what kind of Race is mostly in the database and how that can be considered as discrimination, technological issues, and social implications. Another consequence is using bad samples, such as bad samples that were collected or wrongly collected at the crime scene, causing the wrong information to be interpreted by being degraded, by being different or something like that, and which can convict someone innocent, not the perpetrator. The forensic science uses, there's the four main uses that I was able to find, which is interpretation, differentiate, leads, and identification. The interpretation improves the DNA mixtures by distinguishing alleles. The differentiate is the ability to increase the different individuals by sequencing STR markers. With leads, develop leads from degraded, mixed, and limited DNA samples, and the identification is to identify specific individuals from DNA evidence collected at crime scenes. I'm going more into depth on the identification due to how much more it is. With the DNA sequencing, it allows the scientists to identify the perpetrator or someone closely related to the perpetrator through the processing and analysis of DNA collected at a crime scene. They use it by they use the evidence collected to extract DNA and analyze it, which thus makes a DNA profile of that analyzed DNA. They then put it into a database of other profiles to find a match to be used as evidence.
Kind of going back to the biomedical engineering uses, the three that I have are sequencing, diagnosis, and world health. So going off sequencing, genes are linked to illnesses that now have alterations in their DNA sequences that can be detected. Diagnosis, determining the origin of genetic illness and potential treatments for genetic disease. And on a world health level, it preserves human health and aids in the study of DNA. Also, what is biomedical care? The term biomedical combines biology and medicine to understand and treat health-related issues. So, like I said, we were going to kind of expand on what genetic sequencing actually accomplishes and can diagnose within the sequencing process. And as listed, we have a lot of neurological and intellectual disabilities. We have a lot of frontal lobe dementia. Think like things that are predetermined by your DNA. So the list, I think there's 5 there, there's hundreds. And with more medical research into genome sequencing, we're going to be able to treat these well before and do more preventive maintenance.
With DNA sequencing, there was a project that was created called the Human Genome Project. This was one of the greatest scientific feats in history, and it was led by a group of international researchers looking to study all the DNA of a select set of organisms. It started in the October of 1990, and it's finished of April 2003. The signature accomplishment was generating the first sequence of the human genome, and it provided fundamental information. This project helped accelerate the study of human biology and improve the practice of medicine. The big picture of the Human Genome Project, it helped raise concerns and questions about sequencing, which helped usher in a greater emphasis on the ethics of using DNA sequencing. It produced a genome sequence that accounted for over 90% of the human genome, so that's 90% that we can mostly understand and sequence and know how to deal with it. The project was critical for advancing policies, along with being atypical for biomedical research. Future for forensic science. The future is somewhat vague with being on what exactly would be going on, but what we do know is that the next step would be involving next generation sequencing, which can analyze more genetic markers and provide more information faster, and something like that. Another one, it would be another site states how in the future it may be possible to predict genetic profile without a reference sample based on the known phenotype features of those genes, and how microbial DNA analysis may play a role in placing individuals at a crime scene due to the microbiome that is highly specific to individuals.
For the future in a biomedical standpoint, the ability to sequence DNA within cells is a relatively new technique, but it may open up new avenues for research into the ways which the 3D structure of DNA influences its function and the structural alterations linked to aging and disease, like cancer and brain disorders. It is particularly helpful in the diagnosis of certain uncommon genetic illnesses.
And then these are our references that we use to help figure it out, minus what we know. Thank you for listening to our presentation. Have a great day.